Hey everyone, it's Shane. So for this video we are going to be talking about ATP generation through glycolysis, the TCA cycle, and oxidative phosphorylation in the electron transport chain. Now this is the concept map that I put together that we're going to be looking at. It looks big, but don't really worry about that. We're going to get through it all. And so first off, we are going to start with glycolysis. What we're going to do first is we're going to trace the path of a single glucose throughout this whole thing, and then we'll kind of go off on side trails. So first off, glycolysis, this is all taking place in the cytosol of the cell. So we've got our glucose. It's the enzyme hexokinase is going to take a phosphate group from an ATP and add it to our glucose, and as a result ADP is going to be released, and then our substrate is going to become glucose 6-phosphate or G6P. We'll go ahead and note that if you got an excess of this G6P, your hexokinase is going to be inhibited. So this makes sense. You've got a lot of G6P. Why would you need to make more? Now this G6P can be used as a substrate in other pathways such as glycogen synthesis and the pentose phosphate pathway, but obviously for right now we're going to focus on glycolysis. So the next enzyme in the pathway is our phosphoglucose isomerase. Now it's an isomerase, so it's basically just going to rearrange the structure of the substrate we already have. So it's going to make an isomer, which is going to give us fructose 6-phosphate. Now the next enzyme, you see a lot of stuff going on here. That's going to be phosphofructokinase 1 or PFK1. Again, we're going to take a phosphate group from an ATP, which is going to give us ADP as one of our products. Once this product is formed, this fructose 1,6-bisphosphate, the substrate is now committed to glycolysis. This step with this enzyme PFK1 is also the rate limiting step of glycolysis. And again, this enzyme is regulated. It can be allosterically activated by AMP or fructose 2,6-bisphosphate, or allosterically inhibited by ATP, which makes sense. If you've gotten a lot of ATP, you might not need to make more. And then citrate, which we will see later on. Now next up, we've got an enzyme aldolase, and what this is going to do, it's going to cleave our substrate. So instead of a 6-carbon substrate, which fructose 1,6-bisphosphate is, we're going to have two 3-carbon substrates. One of those is going to be glyceraldehyde 3-phosphate, and the other is going to be dihydroxyacetone phosphate, or DHAP. Now we really want two glyceraldehyde 3-phosphates, so there's another enzyme, triose phosphate isomerase, is going to again make an isomer out of DHAP, and that's going to turn that into glyceraldehyde 3-phosphate, also known as G3P. So now that we've got our two glyceraldehyde 3-phosphate molecules, everything that happens from here on out is happening twice, because remember, we've basically got two molecules at this point. So the next step is, next enzyme in the pathway is glyceraldehyde 3-phosphate dehydrogenase. Now when you hear dehydrogenase, which we're going to have a lot of dehydrogenase enzymes throughout this process, what it's going to be doing, it's going to be oxidizing the substrate and reducing a coenzyme. So you can see here that our substrate glyceraldehyde 3-phosphate is going to be oxidized, which means it's going to lose a pair of electrons and become 1,3-bisphosphoglycerate, and in the process it's going to transfer a pair of electrons in the form of a hydride anion, to NADH, which you can see, and here you've got NAD plus coming in, NADH and an H plus coming off. You've also got inorganic phosphate coming on, which is basically going to be phosphorylating our substrate and going to add another phosphate group on there, which is why 1,3-bisphosphoglycerate is the next product. There's two phosphates. It's no longer just a single phosphate. It's now phosphorylated at carbons 1 and 3. You can see that NADH and H plus is going to go off, but we're not going to worry about that just yet. We'll come back to that shortly. And so now we've got our 1,3-bisphosphoglycerate. The next enzyme in the pathway is phosphoglycerate kinase. Now what you're seeing here is going to be our first example of substrate level phosphorylation, where phosphoglycerate kinase is going to take one of the phosphate groups off our substrate and add it to ADP to make ATP. Now, since this is happening twice, since we have two substrates, our net ATP usage for this process so far is 0, because you remember we spent 2 ATP at the beginning of the pathway to phosphorylate our substrate, and so now we've kind of broken even at this step, and that's going to give us 3 phosphoglycerate, because the phosphate group that got removed from our substrate was at carbon 1, so now we've only got carbon 3 that has a phosphate group on it. Next enzyme is phosphoglyceromutase, and as you can see, the next product is 2-phosphoglycerate, so that enzyme is pretty much moving our phosphate group from carbon 3 to carbon 2. Next up, we have the enzyme enolase, and from this reaction, water is going to be removed from our substrate. That's going to give us PEP, or phosphoenol pyruvate. Now we've got another, as you can see while the thing's going on, another big important step. Another regulated enzyme of pyruvate kinase. It's, again, we're going to take a phosphate group off our substrate, so we're going to, ADP is going to come along, pyruvate kinase is going to transfer a phosphate group from our substrate to ATP. So since it's happening twice, we've now made a net of 2 ATP, so yay, profit. This enzyme, it can be allosterically inhibited by ATP. Again, makes sense, you got a lot of ATP. It won't want to make more, especially if it doesn't have that much ADP available. And then it's also allosterically activated by fructose 1,6-bisphosphate, which was a substrate that we saw much earlier on. It was the one that got cleaved by the aldolase. So pyruvate kinase, now that's going to give us pyruvate. Now there are also other things that can be made into pyruvate: amino acids, alanine, serine, and also carbohydrates. We're not going to get into the really specifics about that. But the next big thing that determines what path our substrate takes is whether or not there is oxygen present. Now we're going to trace the no pathway first, because as you can see, it's very short. So if there's no oxygen available, the enzyme lactate dehydrogenase is going to act on our pyruvate. We're going to go back up here and see the this NADH plus. Now this was going back to our glyceraldehyde 3-phosphate dehydrogenase. It produced an NADH and an H plus. If there's no oxygen, it's going to come down here, and what it's going to do is it is going to reduce our substrate. It's going to reduce pyruvate to give us lactate. Now when NADH is dropping off its electrons, so that's going to oxidize it back to NAD+, where it can go back up there and try again. Maybe there will be oxygen available the next time it tries, maybe not. And then lactate dehydrogenase is going to transfer those electrons to our pyruvate, giving us lactate. That's going to be transported out to the blood. From there, it can go back. It can go into the liver for gluconeogenesis to be turned into glucose, in which case it can go back and try the whole process of glycolysis again. But so let's go back down, and let's say we do have oxygen present. Now here we see our this is our mitochondrial membrane. Granted, yes, there are 2 of them. As you can see, if we go further up, we actually have the 2 layers up there, but for here, since it's just crossing through both layers and going into the mitochondrial matrix, where it's just represented by a little membrane. There you see the lipids, the lipid bilayer. So it's going to usually go to, and then we've got the sometimes go to, but we're going to hold off on that until later. The PDC, that's the pyruvate dehydrogenase complex. It contains components similar to the alpha ketoglutarate complex, which we'll get into a lot later, or a little bit later. Basically, I'd already put the parts of the complex on the concept map for the alpha ketoglutarate complex, and I ran out of room here and didn't want to put it in twice. So we'll just take a look at that a little bit later on. Now, there's a lot going on with this enzyme. It's regulated by lots of things. First off, if it's phosphorylated, it's going to be inactive. So, and it's phosphorylated by an enzyme called pyruvate dehydrogenase kinase. That enzyme is inhibited by ADP and pyruvate. So if you inhibit the inhibitor, you're going to stimulate it. So if you're inhibiting the phosphorylator, the pyruvate dehydrogenase complex, it's probably working. However, if you've got an excess of acetyl-CoA or NADH, that is our reduced enzyme, and acetyl-CoA is our product of the reaction with PDC, it's going to phosphorylate the PDC, therefore deactivating it. Now, if we want to activate the PDC, we dephosphorylate it with an enzyme called pyruvate dehydrogenase phosphatase. That's activated by an excess of calcium. So if that's activated, the PDC is it's stimulated by CoAsh and NAD+, which are products. You can see here, they're going in. And it's inhibited by acetyl-CoA and NADH. You can see those are products. Go down there, it makes acetyl-CoA. It's also going to pump off a CO2, and then it's going to reduce, again, the NAD+ to NADH and the H+, which all these NADHs we're making, we're going to talk about those later on. It's also inhibited by Ley disease, which we'll get into a little bit later because it just breaks things. Okay, so the PDC, it's going to, the main product that we're going to focus that our glucose molecule is now acetyl-CoA. There's a couple of different names for CoA. It's coenzyme A or CoAsh. It's all the same thing. So now we're going into the TCA cycle. Now this is the tricarboxylic acid cycle. You may hear it referred to as the Krebs cycle or the citric acid cycle. It doesn't matter. It's all the same thing. So the first reaction, our acetyl-CoA is going to come in, and it's going to react in the enzyme acting on it is citrate synthase. It's going to take an oxaloacetate as another reactant, combine it with water. The CoAsh that we got from the reaction involving PDC is going to leave, and it's going to give us citrate. Now, this enzyme it's regulated by the amounts of citrate and isocitrate. They tend to exist in a 20-to-1 ratio. If you get too much citrate, obviously the citrate synthase is going to inhibit that. Also, the citrate, remember we had, I think, was it PFK1 way back at the beginning? Yes, is PFK1, the enzyme back in glycolysis, is also inhibited by an excess of citrate. So you'll notice here the green around the enzyme means that the reaction has a negative delta G, which means it's energetically favorable, it's exergonic. The red box means that it is energetically unfavorable, it's endergonic, it requires an input of energy. Now the TCA cycle has a net negative delta G, which means it can happen on its own. The reactions that provide energy, or that produce energy, can then be supplying energy to those reactions that use energy or need energy. So we've got our citrate, which can also be used for fatty acid synthesis. This is just important to know. Next reaction, aconitase, and you see no other inputs here, and it's making isocitrate. So that tells us, since we've got nothing else going on, isocitrate is just going to be an isomer of citrate, which makes sense. If you want to move something around, you don't have anything coming in or going out, it's going to take energy. So I would assume. Next enzyme we've got is isocitrate dehydrogenase. So we see dehydrogenase, what do we know? A coenzyme is going to be getting reduced. So you got NAD+ getting reduced to NADH, and that's going to go to complex 1, which I think I might have forgot to put a label or two earlier on. Again, we're going to get to that later. It's another regulated enzyme, isocitrate dehydrogenase, allosterically activated by ATP and inhibited by an excess of NADH, which makes sense. If you've got a lot of NADH, not a lot of NAD+, you're not going to have a coenzyme available to donate those electrons to. And the electrons, if you haven't figured out yet, are going to be really important in what's going to happen later. So isocitrate dehydrogenase is going to give us alpha-ketoglutarate, which it's also used for amino acid synthesis, and glutamate and GABA, which are neurotransmitters. You can also make alpha-ketoglutarate from glutamate by using either a glutamate dehydrogenase or a transaminase enzyme. So we've got our alpha-ketoglutarate. The next enzyme is our alpha-ketoglutarate dehydrogenase. Now we're going to spend a little time talking about this, the alpha-ketoglutarate dehydrogenase complex. It's a complex containing, well, the three ones that we're going to look at are a thiamine pyrophosphate, lipoate, and FAD. Thiamine pyrophosphate is clinically relevant because a lack of thiamine or vitamin B1 deficiency can cause beriberi and cardiomyopathy. I think they're actually supposed to be tied together. The cardiomyopathy is related to beriberi. The thiamine pyrophosphate cleaves a carbon-carbon bond next to a keto group and transfers the acyl portion from E1 to lipoate on E2 as part of the complex. And then the lipoate contains a disulfide group which accepts the electrons when it binds the acyl fragment of the alpha ketoglutarate and transfers it to the active site containing CoAsh. Then FAD accepts electrons from the liposulfhydryl group and transfers them to bound NAD+. Now I'll go ahead and mention now that FAD, that stands for flavin adenine dinucleotide. And you see we've been making all these NADHs. We haven't really, we'll see FAD a little bit later on, but since it's part of the complex here, I'll go ahead and mention it. It's a coenzyme, but it's attached tightly to its enzyme, whereas NAD, and hence NADH, is not attached quite as tight. It's not really attached to its enzyme. It can pretty much go where it wants, and yeah, that's probably why we see a lot more of them. Binds to a dehydrogenase, gets reduced, and leaves. Well, that was NADH. FAD, again, well, in this case, in the complex here, it transfers them to a bound NAD+. However, FAD can also be reduced to FADH2, which we'll see later. So let's go ahead and go back to our alpha ketoglutarate dehydrogenase. Now this reaction with the alpha ketoglutarate, it's also, we've got CoAsh coming in, we've got NAD+ being reduced to NADH. We're also going to break off a CO2 here. So when we started the TCA cycle, we had 4 carbons in our substrate. We had, I think, we had 2 in our acetyl-CoA that hooked up with oxaloacetate to give us a 4-carbon substrate. And now since we've broken off 2 CO2s, we're back down to 2 carbons again. So again, NADH and the H+ are going to go to complex 1. And the product of that alpha ketoglutarate and dehydrogenase enzyme is going to be succinyl-CoA. You can also make that from valine and isoleucine. The reason I'm mentioning these is they're reactions that can replenish your intermediates in the TCA cycle. They're called anaplerotic. So if for some reason your body's running low on an intermediate, it can throw it together from another way and through some other means. In this case, valine and isoleucine. Succinyl-CoA, it's also used for heme synthesis. So it's important to note that these intermediates, as they're being made and they're being used, they can be siphoned off to do other things as well. So the reactions, succinyl-CoA is going to be acted on by the enzyme succinate thiokinase. So in this case, the CoAsh is going to leave, which, you know, kinase phosphorylating thio-sulfur, so that makes sense because CoAsh, the S, is sulfur. You're also going to have a GDP, that's guanine diphosphate, and your PI, your inorganic phosphate, and that's going to make GTP. So it's kind of random. The GTP, it's important to know that it's made here in this step, but because we're not really making any GTP anywhere else here, I don't think. All right, so the succinate thiokinase, surprise, surprise, gives us succinate. Next enzyme is going to be succinate dehydrogenase, and now here we go with the FAD is going to be reduced to FADH2. We've seen things going to complex 1. FADH2 is going to go to complex 2. Again, we're not going to worry about that yet. The reaction is going to leave us with fumarate, which can also be generated from amino acids. So the enzyme here is fumarase. We're going to bring in some water as well to give us malate, which can also be used in the liver for gluconeogenesis. Now the next enzyme is malate dehydrogenase. So dehydrogenase, yep, we're turning an NAD+ to an NADH, and it's going to give us oxaloacetate, and so which can also be made by aspartate. And we're also going to back up because you can see all this stuff going into oxaloacetate, which I'll go ahead and say now can also be used for synthesis of other amino acids. So remember back here, our pyruvate has oxygen. It usually goes to the PDC. Sometimes it can go to this other enzyme inside the mitochondria, pyruvate carboxylase. Now this enzyme is going to use ATP and CO2, which those are normally things that we want to make or break off. However, this is going to use them. And now this is activated by acetyl-CoA, which makes sense because if you go through the PDC, it's going to make acetyl-CoA. So if you've got a lot of acetyl-CoA, it might just make oxaloacetate, especially if it needs it, if it's running low. So it's activated by acetyl-CoA, inhibited by intermediates. So I'm guessing if you've already got a lot of oxaloacetate, you're probably not going to want to make more. It'll probably rather preferentially make acetyl-CoA. And Ley disease, we're going to talk about this now. So what's going to happen is your pyruvate carboxylase, if it breaks, if it's not working, and again, the same is going to apply to the PDC. If you're only going to make one and not the other, eventually you're going to have so much made of one that it's not going to want to make anymore. And the only other option the only other thing you can make with pyruvate is lactate. So you're going to have too much lactate. It's going to cause lactic acidemia, which will lead to severe mental retardation and death. This is considered a mitochondrial disease, and I'm pretty sure Ley disease is genetic. So we've gone through the TCA cycle, and now we need to go back and look at those enzymes we've been reducing, those coenzymes we've been reducing, because those are going to go to the electron transport chain. So I'm going to go back to the one that we had made here, the NADH that we had made in glycolysis from glyceraldehyde 3-phosphate dehydrogenase, because everything else, all the other NADHs and FADs, or the one FAD, was generated in the mitochondria. However, this was made in the cytosol, and NADH can't just cross the mitochondrial membrane. It's not lipid soluble. So we have got to find another way to get it there, and fortunately our cells have two shuttles, two ways of getting the electrons in there. The first one is the glycerol 3-phosphate shuttle. So what's going to happen is we've got our reduced coenzyme, NADH, and also our H+. It's going to be acted on by an enzyme called cytosolic glycerol 3P dehydrogenase. What that's going to do is take a DHAP. Remember, if you don't remember DHAP, that was one of the molecules that was formed when our substrate got cleaved way back in glycolysis. And so that enzyme is going to oxidize our coenzyme and reduce our DHAP, which will give us glycerol 3-phosphate, not glyceraldehyde 3-phosphate like we had before. It's going to be glycerol 3-phosphate. Now, what that can do is cross the membrane and interacts with another enzyme. This is a mitochondrial glycerol 3P dehydrogenase. Now, it's named just like the cytosolic one, except it's going to do the opposite. Instead of reducing our substrate, it's going to oxidize it, and it's going to take those electrons and use them to reduce FAD to FADH2, which is going to just give us our DHAP back. The DHAP can cross the membrane and be regenerated in the shuttle. The other option for getting those electrons across are the malate aspartate shuttle. This is a little more complicated, but still not that difficult to learn. We've got our NADH and our H+. The enzyme here is the cytosolic malate dehydrogenase. So I'll give you three guesses to what the enzyme on the mitochondrial side is called. What's that going to do? We're going to drop off our electrons to oxaloacetate. Remember from the TCA cycles, this is another thing we could use it for. And the reduction of oxaloacetate is going to give us malate. Malate can cross the membrane. Once inside, oh hey, look, did you get the name right? Mitochondrial malate dehydrogenase. That's going to reduce an NAD+ to an NADH and give us oxaloacetate again. Now, you might have noticed, okay, when it was crossing the membrane, you had an alpha ketoglutarate going the other way, and here's why. So we have glutamate that is, it's just made a trip across the membrane inside of the mitochondria. So this oxaloacetate and glutamate, they're going to transaminate each other. The result is going to be the glutamate is going to become alpha ketoglutarate. The oxaloacetate is going to become aspartate. So I don't know, I'm guessing the oxaloacetate can't cross the membrane. That's the only thing I can really think of, because we see it in the mitochondria and we see it in the cytosol, but we don't ever see it making a crossing. So our aspartate's going to go back across the membrane. Again, we've got another transamination reaction. Aspartate's going to go back to oxaloacetate, and alpha ketoglutarate is going to go back to glutamate, which again you saw it was crossing the other direction of the membrane as the aspartate was. Okay, so now now the electrons are inside the mitochondria where they're supposed to be. That's a good thing. So now we're going to go to our electron transport chain, which is in the inner mitochondrial membrane, because you've got two membranes. The one that's touching the cytosol is the outer mitochondrial membrane. The one inside of that, interior to that, is the inner mitochondrial membrane, and you've got this inner membrane space between the two. Okay, so your NADH is going to drop its electrons off at complex 1, which is NADH CoQ oxidoreductase. It's going to drop them off at the FMN, which is the flavin mononucleotide, which is going to then pass them off to the FES, which is just an FES center binding protein. I like to think of these FES centers as like bus stops, because the bus, coenzyme Q, is going to pick them up and be reduced to CoQH2. Now, from what I've read, CoQ can take one electron at a time or it can take 2 electrons at a time. I'm guessing in this case, since it becomes CoQH2, it probably picked up 2. Though, it could have been 1. No, it's probably 2. Whatever. And so, everything that was going on with complex 1, with the playing of the hot potato, it was like they were playing hot potato with the electrons, passing from FM and FES2. What all that did was it pumped 4 protons, 4 hydrogen ions, from the mitochondrial matrix to the intermembrane space, so across the complex into the intermembrane space. That's going to be important later on. So our CoQH2 is going to go to complex 3. Remember, these structures, they're all 3D, and coenzyme Q is a mobile enzyme. It can just zip around in the membrane, so it's going to go to complex 3. Complex 2 we've got is the succinate dehydrogenase and other flavoproteins complex. That's where FADH2 is going to drop off its electrons. So it's going to drop them off there via the complex's electron transferring flavoproteins, or ETFs. And it's going to transfer them to CoQ, which again becomes CoQH2, and heads to complex 3. Now it's important to note in this complex, there are no protons pumped from the mitochondrial matrix into the intermembrane space. Now that's important to note. No protons here were getting pumped. One of the reasons is complex 2 doesn't span the whole membrane like the other complexes do. So now complex 3, you've got your electrons, well, okay, complex 3 is the cytochrome BC1 complex. You got your electrons dropped off by CoQH2 at the FES center. Remember, we talked about the bus stop. It's going to be passed to cytochrome C1 and then to cytochrome C. Cytochrome C is another mobile protein, mobile coenzyme in the matrix. And that again is going to pump 4 protons from the mitochondrial matrix into the intermembrane space. Cytochrome C is going to go to complex 4, which is cytochrome C oxidase A and A3. It's going to drop them off there, and I'll go ahead and mention now another name for coenzyme Q is ubiquinone. So yeah, it's not a protein, it's a coenzyme. It's not bound to a protein. So anyway, cytochrome, or complex 4, cytochrome C drops off the electrons. It's then passed down to copper complex. There's more than one, so first one copper A, then to cytochrome A, and to cytochrome A3, then to copper B. That's all going to pump 2 protons, not 4, only 2, from the mitochondrial matrix into the intermembrane space. And now this is going to be the site of our final reaction that we have going on. The electrons from the electrons that have been going all over this reaction are going to be contributed here to half of an O2, which is an O, if you think about it, 2 H pluses, and it's going to make water, which makes sense if you think about it, because water is H2O. Those H pluses don't have an electron, so an oxygen atom has 6, but it needs 8 to be happy, like a water molecule. So the 2 electrons that are being passed in from complex 4 can form a single bond between the hydrogen and then to each of, the oxygen to each of the hydrogens. So that gives us water. That's why O2 is the final electron acceptor of oxidative phosphorylation. So we're done. Except not quite. Just like throughout the entire process here, we've been making all these, we've been stripping off all these electrons, and we just showed where they went. So for the last part, we've been pumping these protons into the inner membrane space. And so what those are going to do is go over here to be used by ATP synthase. Now, the pumping of the protons into the inner membrane space is going to set up an electrochemical gradient. Obviously, the inner membrane space, because all the protons are positive, that's going to leave a net negative on the inside of the inner membrane space and a net negative on the inside of the mitochondrial matrix. Additionally, because we're talking about H pluses, we're not just talking charge, we're also talking acidity. You're going to be more acidic, a lower pH inside the inner membrane space, more basic, higher pH outside inside the mitochondrial matrix. So this is going to cause the protons, or H pluses, to flow back in, and there's a pore in this ATP synthase, which is, you can think about it as like a giant wheel or a pump. So the protons, it's going to take roughly 4 to go through the ATP synthase, and what that's going to do, it's going to cause it to spin, and it's going to give us enough energy to create an ATP from an ADP in an inorganic phosphate. And that's really how we're generating most of our ATP. Now, if you want to go back and do bookkeeping, we picked up approximately, I think there were 12 electron pairs that ended up getting shuttled to the electron transport chain. I mean, if you go back and look through, you'll see that it happened 6 times, but remember, from that we had, everything was really happening twice because the one glucose got cleaved. So if it takes 4 hydrogen ions to make an ATP, we had 12 electron pairs going in, and each electron pair would pump out 10 hydrogen ions. So that would give us, what, 120 hydrogens, which is enough to make 30 ATP. Now that number, it varies depending on what textbook you read. The general consensus is somewhere between 28 and 36. 30 is in that range, so we're just going to go with that. And that is it. That is our generation of ATP. Again, there's, especially the electron transport chain, there's a lot of clinical relevance there. Mitochondrial diseases, that sort of thing. So I'd recommend you kind of look into that, especially if you're in med school, because they love testing us on clinical stuff. All right, so that's it. Hope you enjoyed, and I'll see you guys later.